So if you were out yesterday and missed it, you can grab one. But if you have one, it's cool. But you really just need that one for today. I should have asked you to grab my shield while you were in there. It's fine. I'll go. They're actually going to work on a group thing, so I'll go get it.
Oh, okay.
It's cool.
Just because I don't have to carry it all over the place when I'm going to work, I think.
Yeah.
I might be like the absolute slightest.
It's fine. It's fine. It'll be okay. Just think about my office first. See if I'm there, and then go on.
Okay. Sam said they're coming, but they had to ask a question about Chem before they.
Okay. Cool. Thank you. Okay. I feel like I'm missing something. Am I missing something? The chat, maybe. Okay. All right. Is it time? Almost. Almost. Yes, ma'am. The exam one group.
I didn't know if we needed it like just
It should be up there. Look again. It should be up there. It should be up there.
Yeah, it's all the way at the bottom.
Yeah, it should be there. Okay, so I think I made a big boo-boo yesterday. I didn't tell y'all a joke yesterday. I did? I tell y'all two. So then I messed up on my 109 class. It was one class, there was a class that I didn't tell a joke to. So terrible. All right, so do y'all want to hear a great joke about pizza?
Yeah.
Sorry, no, it's too cheesy. All right. So what you have in front of you, what you should have picked up, is this little packet. I really like to do this in groups. I love for people to like put their chairs together and all that kind of stuff, which we're really not allowed to do. So what I want us to do is like take a couple at a time. We're only going to take a couple of questions. Y'all are going to work together. Then we're going to come back together and we're going to go over. Y'all take a couple of questions, work on it, and then we're going to come back together. And we're going to do that throughout this class. Okay. So technically, you're not really supposed to move the chairs and stuff, but as long as it looks like this, exactly like this when we leave, doesn't bother me. But here's the thing. Nobody take your mask down to chat. Okay, I know it's harder to hear with the mask on, but do not take your mask down to talk. Okay, so what we're going to do is take just numbers 1, 2, and 3, just the first page, and as a group, come up with an answer, and then we're going to go over the answers. So if you're on Zoom, you need a handout if you're just walking in. If you're on Zoom, the handout that I gave out just now is, and if you want it on your computer, you can go and get it, is right here. Ooh, it should be up there. Let me move it real quick. Come on. Okay, so it should be, wanted it right. Come on. There we go. Okay. So it's right there, Chapter 8 activity questions. So if you're at home on Zoom, this is what we're going to do right now. So that's what I want you to do. I don't want to give you all the answers. I want y'all to work together, to talk together and figure it out. And then we'll come together and I'll put the answers up as y'all give them to me. What I would suggest, the question is, should we use our notes? Look at it first. Try to answer it without your notes. But there's no penalty for using your notes. But if you don't have to use your notes, you're like, oh, I got this. That means, oh, I have this. I understand this part of the chapter. But if you do have to use your notes, write yourself a note. Ooh, I really need to study this, right? So try without and then do it with. So y'all are welcome to get up and move, you know, to be able to work in a group. You know, it's fine with me. Good. And I'm going to go get my shield so I can take my mask off because I don't know, I forgot. But y'all are just going to work on the first 3, just the first 3. Okay. So go ahead. hang out with your friends so you have less contact with other people, whatever, it doesn't matter to me. But just don't take down the mask. That's all I'm saying. You can turn your chair. Absolutely. Go ahead. Go ahead. Make friends. Okay, I'm just gonna go get my shield and then I'll be right back. How we doing? Okay, anybody on Zoom need anything? Who's on Zoom? Let's see. Never see you. Okay. So I'm gonna pull up the same thing on my flash drive so I can write on it. I just kind of want to see where everybody's at. So it looks like everybody on number 2. So once I kind of see everybody on number 3, you finish at number 3, we'll check. So if you finish before everybody else, go on to the next one. I know that I didn't know if you were starting immediately.
Phosphorylation Cascade.
Would you say the Okay, y'all getting close to wrapping up. 3, maybe. Raise your hand if that's a yes. Okay, go. 3. Okay, keep going.
No, this is the first one. Yeah. Yeah, you took it right. I don't know.
Okay, I think we're getting close. Are we getting close? Yeah. It's okay. So what I'm gonna do is I'm gonna give y'all 2 minutes, and then we're gonna check the first 3. 2 minutes.
Yeah.
And don't forget, it says 22 absent. Don't forget to check in on iClicker. I don't think 22 are absent, but. Take this off. That's why I went and got the mask. So if you finish with these 3, just go on to the next ones, you know, and you'll be ahead of the game. You don't have to wait on us. Okay, so that's our timer. So tell me, A can be a kinase or bind to GTP binding proteins. What is this? A receptor. Can be a kinase, would be a receptor tyrosine kinase, right? Bind to GTP binding proteins would be what kind of receptor binds to GTP proteins? GPCR, G protein coupled receptor, right? Coupled binds to. So what else were y'all saying? So second messengers come after all those adapter proteins. So it can't be second messengers. Right, first messengers are those ligands that are extracellular. What else do we say? It could have, if we had a category that said adapter proteins, could adapter proteins be kinases and can they bind GTP binding proteins? Sure, but that wasn't a category, right? Okay, single pass membrane protein. Membrane protein should have been a red flag, found as a dimer with enzymatic activity. What is that? Receptor. Insulin. First messenger. Cyclic AMP or cyclic GMP? Second messenger. 7-pass membrane protein. Receptor, that's E. Okay, DAG, diacylglycerol. Second messenger. G binds ligand. Receptor. Epinephrine. First messenger. Okay, yeah? All right, questions? Good. Give definition, function, and list examples of each of the 3 categories. So what's a definition of a first messenger? Extracellular ligand binds receptor. That's good. Okay. What else? Induces binding, induces conformational change in, now be careful with this. Should I just say receptor? Intracellular portion of receptor. Now, it could induce changes in other places, but if it never induces a change in the intracellular portion, nothing's going to start. In intracellular portion of receptor. Right? Some examples, throw some out there. Insulin. Give me one that's not up above. Acetylcholine, some more. Calcium. What? Estrogen. Any kind of small molecule. Any kind of small molecule. CO2, cortisol, epidermal growth factor, glucagon, right? Lots of different things. Okay, receptors. How would we define a receptor? Sure, transmembrane protein. Okay. So I would say receives extracellular signal, sure. Receives extracellular signal due to binding. And you could say ligand. Oh, that is not how to spell ligand. Ligand, first messenger. What else? Yeah, it has a conformational change. You can put that. But what's important? So you have this ligand binding to the receptor. It induces a conformational change. So what? So this is the point at which we translate an extracellular signal into an intracellular signal. To translate extra to intra. To intra. TRA, cellular signal. Right? Okay. So some of them have kinase activity. Some of them bind to adapter proteins, right? You could list a lot of different things there that would make that transition happen. So what are some examples? Receptor tyrosine kinase. What's that? Tumor necrosis factor receptor, absolutely. Nuclear receptors, do not forget about those nuclear receptors, right? Okay, second messengers. How would I define a second messenger? Okay, so it's inside the cell. Inside cell. Okay. What's that? Amplifies intracellular signal. So I would say yeah, small non-protein. What are some examples? When we say that non-protein, what are some examples? Cyclic AMP, cyclic GMP. Those are two of them. What else? IP3. What else? DAG. And there's one more we talked about. CA2+. Right? So our signal transduction really begins at what point? Begins at the receptor, is amplified by the second messenger. Right? Okay, what else we want to add? Anything? And what do these things do? What's their job? Bind, regulate, not receptors, or GU, regulate. I would say target molecules. Right? Because they do bind, they do change things, but they don't change receptors. Okay, what are 3 different ways a receptor can initiate signal transduction in the cell? We just said that, right? That the receptor is where the signal transduction begins, and our second messenger amplifies. So what can a receptor do? What is the first receptor that we talked about in detail? GPCR. So I'm not going to say GPCR, but you tell me, what does a GPCR do? When a GPCR binds to a ligand, what does it do? To give me a way that this receptor initiates signal transduction. Does it phosphorylate something?
Why?
It changes binding of adapter proteins, right? So you could say interacts or stops interacting with, right? So causes an interaction or stops an interaction with what kind of a protein? Extracellular or intracellular? Intracellular. Okay, now we talked about RKTs. What do those do? They have enzyme function, right? They have enzymatic function and can act, we'll say act as a kinase. Because sometimes the kinase is going to phosphorylate a different protein, and sometimes the kinase does autophosphorylation of the receptor itself, right? So generally we could just say has kinase activity. Okay, what else? What's that? So usually they're in the nucleus already, right? There are some adapter proteins that in a signaling cascade pathway will dimerize and then enter the nucleus to change transcription. Absolutely. But those transcription factors that are already in the nucleus are nuclear receptors, right? And so those nuclear receptors, so you can put in parentheses nuclear receptors, right? Once they bind the ligand, what are they going to do? They're going to initiate protein DNA interactions, IEN, DNA, and protein-protein interactions, right? I extend the page to change transcription. Because sometimes you want to upregulate something, sometimes you want to downregulate it. It just depends, right? Okay. Anything else? There's another. Yeah, so I would almost leave that under enzymatic function, right? Kinase is an example of an enzymatic function, right? But that's that same thing with the Cas bases. That you're going to cleave it, and that's an enzymatic function. So I mean, I think that fits under enzymatic, absolutely. Well, yeah, that's what he was talking about with the Cas bases.
Oh.
Yeah. So the other one that I thought of is it also creates second messengers, right? So it's also involved in creation of second messengers. Either way, because usually your second messengers are in one state and they're off, and the receptor, when the receptor becomes active, it converts it into the form. So you could say it either way. You could either say creates that second messenger activity or activates it, however you want to look at that. Okay, you ready to go for the next? Do the next 1, 2, 3. Do the next 3, and we'll see how we are. Because that's 4, 5, 6, and 7. Go through number 7. Go through number 7. Okay. Good. All right. So I'm going to give everybody some time. Go 4 through 7. What do y'all think of this? Is this helping? Yes? Okay. I don't want it to be a waste of your time, but
And does anybody need another copy of the abbreviations? I hear lots of abbreviations going around. You know what they mean? Anybody need one? We got them yesterday, but I'm not gonna get another one.
Oh, look, about it, it's Chem. It just has the starters in it, so I don't know. But like, most pressing. Yeah. The way I remember it.
Yes. Oh, like, yeah, it's, yeah, I already got it. So, like, really, do you have one of these else? No. Sad. Any other? No. Oh, yeah. I know.
Okay. So about one more minute, and then we're going to switch over. Ooh. Okay, masks have to stay up, y'all. All right, so y'all ready? It's close. It's close. We get through more of these. All right, number four like comes straight out of your notes, right? So tell me what's this receptor right here? GPCR. Can you describe a GPCR? It controls association with G protein adapters, yep, to initiate signaling cascades. What about the next one? RT-PCR, which has intrinsic what? Kinase activity, phosphorylates what? Tyrosine residues on itself and adapter proteins, right? Okay, so what about this one? TNF alpha receptors, TNF receptors, which control cell survival or apoptosis, right? Depending on what's going on inside the cell, what kind of adaptive proteins you have in the cell. Do you have pro-apoptotic or do you have pro-cell survival? Okay, and the last one, nuclear receptors, right? That dimerize and control transcription through interactions with the receptors themselves in DNA and the receptors and tons of other proteins that bind and regulate transcription, right? Okay, that you can get out of your notes. Okay, what is the purpose of the phosphatase? Not what I.
You took 5 classes.
Oh, and the ion channels. Yes, and the ion channels. Ion channels. And a brief description. So what's a brief description of your ion channels? Yeah, they can absolutely regulate action potential, right? In all of our nerves and our muscles and all that kind of stuff. Yes, absolutely. Anything else? From low concentration to high. Wait, say that again.
It mediates the concentration of ions.
Yeah, it controls concentration on one side of the membrane versus the other. Absolutely. Absolutely. Yeah. Okay, so what is the purpose of the phospho-Anostatic cascade? What's the point? What do we want to generate? What is the name of the game? We want to make this is an example of DAG. This is an example of IP3. Hello. So, we want to make second messengers. And we make DAG, IP3, and we make eventually, which we'll show down below, what's the third one that we make? Calcium, which binds to calmodulin. Make second messengers to what point? Like, why do we want to make second messengers? To amplify, that is the whole point of second messengers, to amplify our signal cascade, our signal, however you want to say that, signal after receptor activation. Right? So what are we starting? What material do we start with? PIP2. So this is embedded in the membrane. This whole thing is embedded in the membrane. Membrane bound. Right? So what enzyme comes in and converts PIP2 to IP3 and DAG? Phospholipase C, PLC, phospholipase C. A lipase is an enzyme that will break down lipids, right? Lipase. And these are lipids, yes.
So which one is the adapter?
They're not. They're not. They're not proteins. They're molecules. You could almost say that this is, but it's not really. It's a substrate. It's really the substrate that PLC was activated by an adapter protein from the receptor, right? So PLC is really your downstream target to become active. This is just the substrate for PLC. So if you look at the cascade, right? Receptor activation activates what molecule? PLC. PLC is the key, right? Because without PLC, nothing's going to happen. So if you have PLC and PLC is active, you're going to take PIP2 and you're going to turn it into what two things? IP3 and DAG. Not an N, DAG. Okay. Now, IP3 can do what? It's going to release calcium to the cytosol. So now you have high levels of CA2+. So how many second messengers do I have here? 1, 2, 3. So once we activate this second messenger, the goal of second messengers is to go on and promote more cascading signaling. So we're going to activate another protein, another enzyme. What other enzyme? What is PKC? You're right, PKC. Protein kinase C. Now we're going to start a phosphorylation cascade, right? So much, right? This calcium can also activate PKC, but in addition to that, it can also bind to calmodulin, M-O-G-U, modulin. And in the presence of CA2+ and calmodulin, you can have some additional downstream effects of, you could just say target proteins. Right? So we just talked it out, but if you can write it, it's going to help you. Okay. All right, so 6. We have PI3 kinase is activated by insulin and other hormones. The insulin receptor, once bound to insulin, leads to the activation of what? Immediately. IRS. That's our insulin receptor substrate. So 1 is D, right? Which in turn activates PI3 kinase to produce PIP3. PI3 kinase is a dimer of 110 kilodalton catalytic subunit and a regulatory 85 kilodalton subunit containing SH2 domains. Binding of the PI3 kinase regulatory subunit to phosphotyrosines induces a conformational change of the catalytic subunit, while blank association. So are we going to induce or are we going to disrupt? We'll maintain or disrupt, I guess, put it that way. You're going to keep associating with the regulatory subunit. You're not going to lose the regulatory subunit. You're going to still keep the regulatory subunit. So once we're phosphorylated, right? Once we have the proper phospho-Anostatic generated by PI3 kinase, the lipid is going to be an anchor for what? Well, so anytime you have something that's phosphorylated, right? Now it's a new docking site. Things can't dock, things can't bind if they're not phosphorylated, right? Otherwise, you would have signaling going on without receptor activation and downstream phosphorylation events. So those phosphorylation, those proteins, once they're phosphorylated, now can bind to new things. So what should 4 be? the pH binding. Okay, now PI3 kinase leads to increased glucose uptake and glycogen synthesis to, what do we want to do to our blood glucose levels? We want to lower them. We want to lower them, right? That's what insulin does. It says take in glucose. Okay, so anybody get to this one? You want to give me your order? Anybody got this one? Anybody? You don't want me to give you the answers? But like, you're gonna try it, right? Okay. So 6, 5, 7, 8, 4, 3, 2, 1, 9. And there are some extra questions, but it's fine. I feel like maybe I'd give the answers tomorrow. I want you to try these. I guess I'll post the answers. Somebody send me a reminder email and I'll post the answers tomorrow. Did you swipe up?
No, I'm saying, you may need to remind people. I don't know if you.
I did. I
Okay.
did remind people. I said there are like 22 people absent. I don't think there are 26 people absent. If you need to do your eye clicker, do your eye clicker. Was it helpful?
Yeah, I actually really like that.
Yeah?
It really made me like focus on what I'm writing. I'm
Yeah.
not just taking a note.
Yeah. You're thinking.
Yeah.
And that's kind of how you should be studying.
Yeah, that is what I do. I go in the textbook, I go in the questions in the back of the textbook, and then I try and answer them as if it was a test.
I love that. Yes. I had trouble finding the abbreviation chart on Moodle. Did you find it, Tammy? No. Okay. Let me see if I. Let me look. Hold on. Do I? Did I put it in the wrong place? Okay, Tammy, when I get back to my office, I am going to post it. And when I post it, I'm going to put it right here underneath activity questions so that it's easy to find. Oops. Trying to look at my chat now. Where did my chat go? Here it is. Okay, so I'm sorry. I don't know where it ended up, but I will repost and I will put it in there. And it's also, it is 2 of your tables if you want them right now. It is table 8.3 and table 8.4. It's those 2 tables. And I will go ahead and post it.
I'm studying it. You look for my email.
Okay, sounds good.
Dr.
All right.
V.
So if anybody else has any questions, everybody good? Okay, I'm gonna end the Zoom.
Fourth exam in 2 days, so my brain is shot.